Hi everyone, my name is Kang Jin. It's my honor to be here to present our recent tool, CellDrift. And I'm a PhD student from Cincinnati Children's Hospital, and my supervisor is Dr. Bruce Arenal. And the tool, CellDrift, is about the inference of perturbation responses in temporally sampled single-cell data. And this is our bioarchive preprint. So I want to talk a little bit about our background and motivation to develop this tool, because nowadays single-cell data is more and more commonly seen, and especially with like the reduced of the cost, we see large-scale single-cell data, and a lot of them are actually perturbational data. For example, this one talks about the immune responses after the influenza vaccination, and the next one talks about the neural development in the brain organoid with the autism risk gene mutation. And the third one talks about cell lines with a kidney disease during the organoid development. And all of those three data sets are about temporal perturbational data using single cell sequencing. And just a cartoon visualization about such data set, which include both perturbation conditions as well as temporal data. For example, the single cell data along the disease progression, like the kidney disease here in the third paper, and also like the cell type profile during the organ development as well as drug responses. And this is another visualization that we can just show those data within like a time series, and in each time point, we can have a combination of different cell types and perturbation groups. And this is like the complexity of such temporal perturbational single cell data. And before we go into the detailed method, I want to talk a little bit about one of the well-established longitudinal analysis package, or not package, area called functional data analysis, also called FDA. And it can be used in a lot of ways. So it's a very versatile method or area. For example, this one is temporal curves for the weather, the temperature recordings in Canada, and there are a lot of curves. Each curve represents one location where the temperature along the year were recorded. And for those different locations, like each location has a temporal curve, and we combine those temporal curves to conduct functional clustering. We can see like three categories of the clustering represent three like geographical type of weathers in Canada. This is like one kind of application using functional data analysis to do the unsupervised clustering. And another type of application, which is also very popular in functional data analysis, is functional PCA, where like you can reduce the complexity of temporal data, because like in very complicated temporal curves, there are a lot of variations along the temporal dimension, and you want to reduce them into several like lower dimensions in similar to the normal PCA, and the functional PCA is an option to reduce that temporal variation complexity. And actually, there are a lot of like different functions that are available in this like scikit FDA, which is in Python. And for example, clustering and classification, extrapolation, functional PCA, as well as like functional ANOVA can be easily achieved using this package. And this is also one of the basic package that we use in CellDrift tool. And so how can we represent data in functional data analysis? And this is an example of glucose levels of these patients. And for each patient, the glucose level during the day were recorded, and it was recorded for 7 days in a week. And those four figures are actually for patients. And actually, the functional data is very simple. It's just a definition of a measurement during the time. So a time curve, temporal curve, is a functional data. And if you have temporal data from different samples, that's functional data. And you can easily conduct functional data analysis using different functions to analyze this temporal data. So this is what we use. And additionally, another background is the generalized linear model. And this is like a linear model, which is a very simple regression model. And in generalized linear model, people introduce like link function in combination with probabilistic distribution, like Poisson distribution or negative binomial distribution to mimic count data. And as we know, like bulk RNA-seq data or single-cell data are just count data and can be perfectly modeled using the generalized linear model. And here are some popular application in bulk RNA-seq or single-cell RNA sequencing, like DSeq2. And we kind of borrowed advantage of generalized linear model to resolve the perturbation effect in single-cell data. So this is a workflow of CellDrift. So as we mentioned, like we have on the panel B, we have different like temporal perturbational data. And within each time point of those data, we can see a very complicated combination of cell types and perturbation groups. So our approach is to use generalized linear model in combination with functional data analysis to resolve these complicated dimensions of the temporal perturbational data. And our ultimate goal is like what we show in panel D. We want to identify different patterns like treatment A, which has a very distinct temporal perturbation responses compared to treatment 2 and treatment 3. So they can, like, for example, if we apply temporal clustering, we want to identify these two very distinct patterns. And our approach is to use generalized linear model to decompose the single cell data and extract the perturbation effect for each single time point, and then concatenate different time points and input that into functional data analysis to conduct analysis like temporal clustering, functional PCA, or functional ANOVA, which were all achieved in CellDrift package. And this is an overview of our model. And we model single cell raw data, which is count matrix using gamma NG, and N is cell N, G is gene G. And we measure the average of the negative binomial distribution for count data using a linear combination of a lot of covariate, including like technical covariate like size factor, intercept, and batch effect. And we also measure other biological covariate like beta GC, which is a cell type coefficient, and beta GP is a perturbation coefficient. And in addition, we also make it into an interaction model, which means we consider about the interaction between cell type coefficient and perturbation coefficient, and we make it as beta GCP. So basically, we want to use beta GC to measure the common perturbation coefficient, which is shared between different cell types in response to a specific perturbation. And beta GCP is more cell type dependent. That's to say, different cell types may respond differently towards the same perturbation condition. And this is the general model in measurement of the perturbation effect. And actually, some people may wonder what's the like real effect after adding this interaction term beta GCP. So actually, we looked at the reference model where we didn't consider the beta GCP versus the contrast model where we consider the cell type specific perturbation beta GCP. And we use the likelihood ratio test to see if the addition of this covariate can make the model more robust. So we actually look at the likelihood ratio test result, and you can see in panel A, they are the results that are not significant for such test. That means for different cell types in the x-axis, their perturbation responses, which is evaluated in the y using contrast coefficient, they are not different. And in contrast, we can see like the significant likelihood ratio test in panel B shows a significant difference for the perturbation responses between cell types. So for some genes, we really want to like add this term to make the model more robust and more specific to such detailed perturbation effects. And since we have like different covariate, we kind of like statistically combine the beta GP and the beta GCP and make it into a contrast coefficient, which is a representation of perturbation effect in single cell data. And actually the covariate is calculated for each time point. And after we manually concatenate the result from all the time point, and we can make it a series of temporal perturbation effects. And we define this series as functional data. And this functional data of perturbation effect can be used to in two aspects. One aspect is that people can like concatenate different genes for a specific cell type and perturbation group and see what's the temporal variation between different genes. And we define it as FG. And some people may be interested in looking at the variation of a specific gene across different cell types and perturbation combinations. And this is also enabled using this definition in the second algorithm. And this is the coming back to the workflow. And with this method established, we want to benchmark how it performs. So we benchmark actually two parts. One part is the generalized linear model part, and the other one is the functional data analysis part. And for the generalized linear model, we want to see how good the CellDrift model performs to identify perturbed genes. So we simulate perturbation data using splatter with different batch effect, as well as different differential expression size, which is not shown here. And we evaluate, we also define the ground truth, and we compare our predicted significant gene versus the ground truth. And we found that across different levels of batch effect, CellDrift always performs best in the true positive rate. However, the false discovery rate is not always the best, but it is stable across different batch effect size, similar to MUST, probably because that we take the batch effect into account in our generalized linear model. And the other benchmark is looking at the TPR and FDR in different differential expression size. And again, the TPR is pretty good, outperform other methods, but still we saw some limitation in the FDR measurement, and that can indicate some limitation of the method. In addition, we looked at the benchmark result for the temporal pattern identification. That's to say, we simulate temporal perturbation effect, and we want to see whether CellDrift can identify the temporal perturbation effect more accurately than other methods. So we simulate this simulation is actually I borrow some ideas from this Nature method paper, which is also about the temporal analysis of single cell data. And we built 3 temporal patterns, and each temporal pattern indicates different perturbation responses. Pattern 1 is positively related with time, and pattern 2 is time independent, and pattern 3 is negatively correlated with time. And we simulate data in different parameters, including different time point and different ratio of missing time point and different noise levels. And in addition, we also simulated more complicated nonlinear temporal patterns to mimic more complicated real temporal perturbation effect. And we benchmark our strategy of CellDrift, which is the combination of generalized linear model output with FDA clustering method called fuzzy C-means. So that's our CellDrift approach. And we also benchmark it with other alternative approach. And we use the ARI to identify whether we can accurately identify the temporal pattern that we defined as the ground truth in our simulated data. And we can see, although that our strategy is not always the optimal one, but it performs quite stably and in a pretty safe range compared to other methods, and not pretty disturbed by all kinds of parameters. And we also looked into the imputation of temporal perturbation effect for the missing time point, and our method significantly outperform other alternative approaches. And with the benchmark result, we want to see how it performs and how it can be applied to the real dataset. So actually, we applied it to a large-scale COVID-19 example, where you can see from the x-axis, there are 6 conditions, including 2 mild COVID-19 condition, 1 severe COVID-19, and 1 critical COVID-19. In addition, we also include 2 other infection diseases, including flu and sepsis. And we extracted 6 commonly seen cell types in PBNC data of this COVID-19 patient, and we recorded their time point, which means the days since disease onset. It ranges from day 0 to day 25, representing early stage of the disease progression and the late stage of the disease progression. And we applied CellDrift to one of the cell type, which is classical monocyte in the severe COVID-19 for different genes. And we found that, so I want to mention that the black curve is the average curve, and we identified 20 clusters, and we show 3 of them, which is cluster 11, 13, and 17, and the cluster 11 is like in the beginning lower than average, which means like it was suppressed in the beginning and then recover in the later time point. And in contrast, the class 17 is upregulated in the beginning and then go to normal in the later time point. And we do gene enrichment analysis and find that actually cluster 11 is associated with biological metabolism, and cluster 17 is associated with immune responses, which makes sense because it indicates that the genes of housekeeping genes were suppressed in the beginning, but in contrast, the immune response button will open after the virus infection, which was validated by previous literature. And this is a very simple validation or sanity check. And we also validated our patterns using external gene patterns from literature. And to save time, I will just skip this part. And the conclusion here is that we find that actually that our temporal patterns that are more upregulated is more associated with critical COVID-19 modules in the external literature. And our more downregulated patterns is more associated with control modules. And we also validate our approaches across different cell types, because previously that's more about one cell type. And we looked at different cell types and identify very distinct patterns. And the gene enrichment result shows that actually the upregulated temporal patterns and downregulated temporal patterns can be associated with very distinct enrichment. We also applied ANOVA test, and to save time, just sorry about that, to skip this part. And we also validate the application in serial time, and it works as well in organoid data. And to conclude, I want to say that we developed a generalized linear model to decompose the temporal perturbation data and find some patterns that are associated with real biological indication. And there's still some limitation, including we didn't consider the time as a covariate in the generalized linear model, which limit our exploration of covariance between time points. And with that, I want to thank my team, my PI, Bruce Arano, and other people who is involved in this work. Thanks a lot. And with that, I'm happy to take any questions. Thanks.
So thank you a lot.